First of all, I would like to thank Tema for the nice organization of this event and also, of course, for the opportunity to talk today and to share with you the latest innovation of our high-speed inspection technologies. My name is Sven Breichert. I'm working for Pharma Technology almost 2 years now. Today I would like to talk about Q-Controls. Q-Controls is one of our new developments. So it's a very high-speed inspection. So we will learn a lot today of how that works, what are the technologies inside. And I'm very lucky, so I can do 2 presentations today. So I split the presentation first in the technologies, how that works, what machines do we have, and the second one is really based on case studies. Maybe 2 sentences to pharma technology. So we are now in the market since almost 30 years. So we are manufacturer for OSD, oral solid dose products. And the headquarter is in Belgium, very close to Brussels. Good. Now let's talk about Q-Control for 100% inspection. So you see here the 3 machines. What we are going on. I hope this is the right one. Perfect. So we have here our CAP 20, we have a CW 150, and we have a CU 120. First to clarify about the numbers, so this represents the maximum throughput. So we are talking about 20,000 tablets per hour, what can be inspected by the CAP 20. We are talking about 150,000. Here we can inspect capsules and tablets by the CW, and we're talking about 120,000 of the CU 120. Let's start with the CAP 20. So it's the first machine here. So this is a benchtop machine, a very versatile machine. It's designed for process understanding and for process design. On board, it has laser scanning technologies to measure the thickness of each individual tablet, to measure the diameter or the length if you have oblong tablets. And on top of that, we have the next station, an NIR station, where we can measure API fractions. I will later talk about application examples. Going to the CW 150, this is a 100% check weigher for tablets and capsules. So you can easily change between capsules and tablets by changing format parts. Here we can go up to 150,000 objects per hour. The technology inside is a 3D microwave, what is here in the heart of the machine. So 3D microwave will give us the mass of each individual object. And then at last, we have our CU 120. So CU is content uniformity. So here we have a mixture of everything. So we have laser scanners, we have our NIR, and on top of that we have our 3D microwave, meaning that we can measure the thickness of tablets, the diameter, we are measuring the API fraction, and at the end we are measuring the mass. So we can marry API fraction with mass. So we have the unit dose, the milligram API, what is nothing else as the content uniformity. Before I dive now deeper in the machines, I have 2 slides about PAT and quality by design. So we heard also from Bruno today a lot of CQAs, CPPs, so process parameters, control attributes, quality attributes, and so on. So I would like to skip most of that. So you see also here our nice quality by design. You see I usually read that from here to the inner circle. We need a control strategy, we need our quality attributes, we have our process parameters, we have our inspection technologies, and then we come to our quality by design. More easy is it just to make an example. An example, for example, a tableting press. So, of course, on top we have our product quality, and if you have our product quality, we have to define our CQAs. Examples for CQAs could be content uniformity, maybe hardness, because hardness is very important for bioavailability of a tablet in the stomach or whatever it have to take an effect. So what we have to do is we have to do a DOE, a design of experiment, where we have to link our CPPs, our process parameters, to our CQAs. So for example, compaction force to hardness. We have also seen this today. Okay? And if we know that, we perform our risk assessment, we can develop and define our design space. We can implement our control strategy to really know what's going on in our design space, to survey if we are really inside the design space. And of course, the latest step is here the continuous improvement. So as an example of that, we have here, for example, the turret speed, the compaction force as process parameters. We have our CQA, the hardness. So that means we have here our design of experiment. We have the design experiment much larger than the design space. And of course, the control space must be the smallest in order to guarantee that we are always within the design space. Okay, talking about enough about that. Let's now jump a little bit into sampling in batch process. So we see that the batch release is dependent on different parameters. We have mass, uniformity of mass, we have our content uniformity, and we have others like dissolution, impurities, and maybe some others, depending on the product, depending maybe also on the country. Let's now focus on content uniformity. And here it's written in the US 905, or also for Europe in the FARM Euro 2.940, that we have to take 10 tablets from an entire batch, and we have to do our assay of sample. And now, my question to you is this sufficient? If we consider a batch from 500,000 tablets, if we consider maybe a batch of 5 million tablets, can this be sufficient only to measure this small amount of tablets? For my opinion, no, it cannot. And so we have one of the main drivers of our machine. We can do 100% inspection. 100% inspection of content uniformity. That means you have really an idea what's going on in your process all the time, in line. A second point is the time after finishing a batch until the batch can be released. I know you can probably prioritize, but usually, I can tell you what I heard, that usually we need 2 to 4 weeks until the batch is ready, until the batch is released. And this is a lot of time, and time is money. So this is the second driver of using our machines. You can apply for a real-time release. We have the mass of each individual tablet. We have the assay, the content uniformity of each individual tablet, and based on that, we can release the batch. So we are talking about real-time release. Let's now focus on the free machine itself. So I promised that I will give you some application examples. So let's start now here for the CAP 20. So remember, we have our laser scanners, we have our NIR on board. So we can drive, or we can use the machine in 3 modes. So we can use it offline, we can use it at line, and we can use it online. So for online, of course, I told you, it's more for process design, it's for process understanding. You see, the machine is very versatile. We are on rollers, so you can bring the machine from one room to the other. So we can use it for research and development, and as a PAT toolbox. With this machine, you can very easy and very fast collect spectra. And now I have maybe 2 examples for you how this works. For example, if you are very familiar with NIR spectroscopy, near-infrared spectroscopy, you know how difficult it can be to find the best measuring setup. So you have to find the best integration time, you have to find the best distance from the tablet to the probe, and so on. This, for example, is completely embedded in the software. So the software will ask you just to provide 5 to 6 tablets. The tablets will be driven under the NIR sensor it will stop, and then immediately spectra with different integration times plus different distances from the tablet to the probe are taken. The best result will be calculated and automatically stored in a recipe. Of course, if you have more knowledge, you can change that, but usually it's a good idea to use that. A second parameter is the design of experiment, what is also embedded in the software. So you just need to tell the system, okay, for example, I have 3, 4, 5 different dose strengths of my tablets. I have, for example, 20 tablets of each, and then the DOE is automatically calculated. And the software is asking, please provide, for example, the 85% tablet number 3. So you can just provide it to the bowl, it will be measured, and then you get a call, you have to go to the boss for a meeting. After 2 hours you come back and you have no idea, oh, where did I stop my measurement? The system will let you know. So the complete DOE is embedded in the software. On top of that, of course, we can use the machine for proof global batch quality. We can also detect counterfeit products. So you have a lot of applications. In add-line mode, of course, you can allow stratified sampling method and you can improve the real-time decision. And of course, you can also serve multiple lines because the machine is very versatile and transportable. In an online mode, you can use the machine for the automatic sampling at regular intervals, but here we have the limitation of 20,000 tablets an hour. So probably it cannot follow full production speed. Let's now take a look on the functional blocks on the machine. So usually you fill bulk material, bulk tablets into the machine. So we have a hopper here. The tablets will be singleized. So we have a centrifugal bowl, what is singleizing the tablets. This is very important. And then we have a transfer from the tablet, from the centrifugal bowl on the belt. On the belt, the tablets are transported and held by vacuum, so it cannot move anymore. And then the tablets will be transported first to the laser scanner. So again, tablet thickness, tablet diameter for round, tablet length for oblong tablets. And we are going here for the NIR spectrum to get our API fraction. After that, we have a sorting gate. So we can just say, okay, we accept conforming tablets, or we reject non-conforming tablets. Of course, we have also a confirmation sensor based on capacity that we can ensure that a non-conforming tablet was really rejected. So you cannot have false acceptance. Okay? Going to the CW150. So here we are talking about the checkware for tablets and capsules. We can use the machine for inline and offline. Inline is clear. We use the machine for 100% weighting of tablets or capsules up to the 150,000 per hour. We can also proving final quality on batch or for continuous manufacturing. It's also clear. For offline mode, we have also one very interesting application identified. So we can use the machine as a kind of batch saver. So let's assume QA doesn't release a batch, too many outliers. So here in terms of mass. So this machine is going for mass only. It's a checkware. Don't forget this, please. So what you can do, you can bring your batch in the inlet hopper. The tablets will be, or the capsules in this case also, will be singleized. Everything will be measured. The bad non-conforming products will be rejected. With the remaining, you can repeat your release strategy, knowing that all the non-conforming product is gone, and probably you will save it. It is very important if your product is expensive, because then you might have a very fast payback of the machine. Also, to be honest, you cannot do that often. Of course, you cannot do it always, because then the inspector from the FDA is probably coming and saying you are doing quality by sorting and not quality by design. But at least 3-4 times it is allowed if you are just showing that you try to find the issue in the process, and this could be a payback of a machine. So use it as a batch saver. Let's also take a look on the functional blocks of this machine here. So also here we have singularization. You see the setup is a little bit bigger than the other one. So you have an inlet hopper, you have the centrifugal bowl. Here we have singularization. The capsules or tablets will be then sticking here to the belt. Then they will be air injected, so they will be agglomerated up to 10 meters per second. So the products are really flying through our 3D microwave measuring chamber here. I will explain later in detail how this works. And then it will be measured. So again, we can do both, tablets and capsules. Our 3D microwave has an accuracy of plus minus 1 milligram in a 3 sigma process. After that, we have our high-speed sorting gate. This sorting gate is capable to handle up to 45 objects per second. So it's very fast. So we just accept good tablets, conforming tablets or capsules. We reject non-conforming products. And on top of that, we can, I suggest to do it once per minute. Yeah? We can sample one object, so one capsule or one tablet, to the external scale here, what's also in the top of the machine. With that, we can ensure that any changes in the humidity, in the temperature, don't affect the microwave measurement. You know that, for example, moisture can affect microwaves. So, and this is what we are doing. We're just measuring here the product. For example, the prediction is 100.5 milligram. Then we direct this product here to the external scale. We measure the real mass, and then we can compare. And if there's the deviation, we can apply our calibration. I will also explain later how this calibration works. But at all moments, you know, the predictions of the 3D microwaves are correct. We know that we have already some existing checkwares in the market, so I would like to point out our advantages. So we do tablets and capsule by changing the format parts. We can upgrade our CW150 with the NIR technology and the laser scanners, of course, to become a full CU120. And then you can go for content uniformity. Okay? And of course, we have here already standard OEB 3. We are working on 4. Today I confirm 3. Okay? Then let's go to our, let me say, high-grade machine. So this is our CU120. So also here we can use the machine offline. So we can use all the functions what we have learned already from the CUP20. So CMOs, we can use counterfeit products. We can also sampling measurement on mass, thickness, hardness, length, diameter, because we have here on top a 4-parameter tester included in the machine. Much more interesting from my point of view, of course, is here the inline mode, where we can go for 100% mass and API fraction inspection. We go for 100% tablet thickness and length or diameter measurement of your tablets. But I just forgot the accuracy of the laser scanner is approximately 0.1 millimeter. Okay? Let's take a look on the functional blocks of the CU120. So singularization, what you learned already, laser scanner for tablet thickness and dimensions. Then we come to our NIR. After that, we have the agglomeration of the product. So we have then the microwave measurement for the mass. After that, we have our fast sorting gate. After that, we have 2 cyclones to disaccelerate the products, so we don't have a tablet crushing machine, that they are cleanly either collected in the bin, also in the goods, or reject bin. And of course, we have here in the bottom our physical measurement station, what I always call a 4-parameter tester. Very important. So we have NIR measurement for API fraction. We have 3D microwave measurement for the mass. We can marriage that. And then we know what is the milligram API in the tablet. And this is the content uniformity. Good. Now let's dive into the technology. So first, let's have a look on NIR technology. You might have read already the term SRS. So what is SRS? So SRS means spatially resolved spectroscopy. This is the technology what we use. So how does it work? We have polychromatic light. That means light from each wavelength. So this light is then provided by these illumination fibers. So this is an illumination fiber bundle consisting of 21 illumination fibers. So it's like a pattern. We illuminate the tablet under 0 degrees. You see that here. The radiation, the NIR radiation, is penetrating the tablet roughly 1 millimeter. It is collecting spectral information of the tablet. Then it is reflected back to collimation fibers, and here we have 9 different collimation fibers. Here you can only see 6, but if you see on the outlet of the probe, you see that we have 4 here, 5 here, so 9 in total. Okay? The next thing is, we are not using a traditional NIR here. So we are using an HSI, a hyperspectral imaging camera. The hyperspectral imaging camera has a detector where you have a spectral resolution. So this ends up here in the 950 to 1650 nanometers. On the other hand, we have a spatial resolution, a locally resolution. What ends up in the 9 collimation fiber. That means if I just apply one image, I have already collected 9 completely independent NIR spectra. And you see also that the travel distance from the light to these different collimation fibers is different. That means we collect a lot of variation of the tablet, what we need for the model creation. And just if I compare this to traditional NIR, it gives us much more information, and this means we can usually increase our accuracy. Okay? Then there's one more thing to tell about the laser scanners. So laser scanners, they care about the tablet height, also the thickness, and the dimension. We talked about that. On top of that, the laser scanner knows exactly when is the start and the end of the measurement. It knows the band speed of the machine, so it can calculate how many triggers, how many images per tablet are possible. And if you see, for example, here this oblong tablet, we are able to apply 5 images on this tablet. So this is just an example. It's not always that this is possible, but in this case, with the band speed, with the size of the tablet, we were able to apply 5 images. 5 images, 9 collimation spectra each. So we collect 45, you can see this here, 45 completely individual NIR spectra. So we have a lot of variation in our spectra, a lot of information. So what we are doing, we are calculating the mean, and this mean spectrum is then predicted against the spectrum. So this is the way how it works. Last thing about NIR, usually I'm facing the question, what is necessary, how is it done with the model creation? So what do we need? We need tablets from different API strengths. So for example, from 75 up to 125%. 75, 80, 85, whatever. Then we are taking the spectra of these tablets. I told you already how easy this is with our design of experiment software. Then either we give these samples to the reference analysis, to the HPLC. So once we have to do this job, at the early beginning we can also take the label claim. But better, of course, is the real values from the HPLC. And then we calculate our model. So usually we are working here with partial least square. This is the most known, and let me say the most used algorithm in chemometrics today. In a second step, we take unknown samples, we measure the spectra, and with the support of the PLS model, we predict API fraction. So this is the way how it works. Are these results within specification? We can validate the model, If not, we have to optimize. Maybe we have to measure some more spectra from one dose strength, whatever is necessary. Maybe we have to apply some different data pre-treatments. There are many ways. But usually this is the way from calibration and validation of NIR models. Good, I promised to stop here with NIR, so let's now move to free microwave. So first, what is it? So we have here a machine PLC. We have CPU, the generator, and the detector. This is what we have 3 times because we are talking about 3-dimensional microwave. We have 1 tunnel. So this is the measuring chamber. We should our sample through this tunnel. So it's really high speed with 10 meters per second. And during the movement of the object through the tunnel, it is measured. Second thing is, these were our first conceptual design. This was the first prototype sensor. And this is now the final result, the GMP sensor with easy to clean, not sharp edges, and all what you usually ask us to do. Good. How is now the thing going on with the measurement? So in the measuring chamber here, we apply our 3D microwave fields. So let's just say this is a kind of 0 state, okay? So the tablet is coming. It is then in the next situation, it is in the measuring chamber. And what is done is that the microfields, they are deflected, they are changed. And this is what we record. We have the 0 state, we have then the changed state, and this difference is what we record. What happens? So the microwave interacts with the dipole of the molecules, and this is the reason that they will be changed. Next position, the tablet or the capsule is leaving the chamber. So the fields are re-established to the 0 state, and they are waiting for the next tablet to be measured. What will be changed? What can we see? So there are 2 parameters per dimension what we record. One is, so we have a frequency shift. So you see this is the 0 state. You see here the resonance phenomenon. You see if we have a tablet inside, we change the frequency. So it's a frequency shift. The second is, you see also that the resonance width is changed here. So the curves are getting wider. So if I have a very large, it's easy, even not wider here. You see that the resonance width is increasing. So these are the 2 parameters what we are measuring per tablet. And now it's also very easy to understand how the calibration of microwave model creation works. So we just take 20 samples. We provide these 20 samples through a microwave. We measure then from these 20 tablets 6 values per tablet. And then we direct the tablets to our external scale. And we measure the real value. And what's done, we just apply the mass, what is provided by the weighting cell, against these 6 values, what we get per tablet. And we apply that in a multi-linear regression. And what we get out is the kind of regression line, and this is our model. And this is also what will be changed during production. Meaning, if we direct this 1 tablet, or for the CW, 1 capsule per minute on the scale, we just compare, and we are just looking for a little bit of the slope of this here, of the calibration. So this is a rolling, a maintained calibration to ensure that we always are measuring the right mass of the product. Time over? Almost. Thank you for your interest for the first one. I'm open for questions, and I promise I will come up later with some nice examples.
Yes.
Okay.